Hi, in this audio lecture, we're going to do an overview and introduction of food microbiology. And first, we'll start looking at microbiology, and then more specifically at food microbiology and some of the history of food microbiology. So, when we talk about microbiology, it's a study of living things that are too small to be seen without magnification. So, we would need a microscope in order to see it. There's some caveats there, and we'll talk about that when we talk about what are microbes, what are microorganisms. Within microbiology, there's 4 different branches. Medical microbiology, that's probably the one you're most familiar with as you've taken an intro micro course, and that would have covered a lot of the medical stuff because we have our allied health and nursing students taking that course also. So traditionally, that's more the focus of microbiology. We also have agricultural microbiology, industrial microbiology, which crosses over into biotech, and then food microbiology. So what is food microbiology? It's the study of the role that microorganisms or microbes play in food spoilage, food production, food preservation, and foodborne diseases. And so we'll be looking at all those components throughout the semester, and what we've done with the course is kind of break up them into the 2 different units, so that we'll be looking at fermented foods in Unit 2. Unit 3 will be foodborne illnesses, and then we have some miscellaneous topics that we'll end up talking about and focusing on, like careers in food microbiology at the end of the semester. So just in case it's been a while since you took intro microbiology, what is a microorganism or microbe? These are microscopic organisms, and commonly for terms like germs, viruses, agents can be used interchangeably in lay articles. And as a microbiologist, I see sometimes I see papers or news articles, and they're talking about someone who's got, a bacterial infection, but then they're talking about a virus, and it's just, so we do want to be mindful that we are careful with the terminology we use. And so microorganism, microbe, is an overall catching term that covers all different types of microbes. Okay, keep in mind that with microorganisms, a lot of times the focus is on the disease that they can cause, or how they can spoil food, but there's also lots of beneficial things they can do. So in an intro microbiology class, you would have talked about your normal flora, your normal microbiota, and how it's actually beneficial to you. It protects you, it helps your immune system become educated, it also provides you with nutritional benefits as far as digesting your food and production of vitamins. And then also when we look at food microbiology, we can utilize certain microbes to actually help preserve foods, and this is done with the fermented foods. So there's a lot of beneficial things that microbes can do, so it's not all bad. So looking at the major groups of microorganisms, they fall into several different categories: bacteria, algae, protozoans, hemolyphs, fungi, and viruses. So bacteria, algae, protozoa, hemolyphs, and fungi are cellular. So they're going to have cell walls, they're going to have structures inside of them for bacteria. And for our eukaryotes, like algae, protozoas, hemolyphs, and fungi, they're also going to have organelles inside of them. Okay. With viruses, those are non-cellular, and they're parasitic. They need a host cell in order to replicate. So basically a genetic component that has proteins coating them, and they're able to infect all different kinds of living organisms, so not just humans. So viruses can be an issue when we're looking at food that is plant-based, right? Because there could be viruses being introduced through that, and also that could damage or denature the produce. So we'll definitely be talking about viruses this semester. We'll be talking about bacteria. For algae, it's going to be more when we talk about like edibles, not those kinds of edibles, but edible microbes. So some of these microorganisms are actually like seaweed, right? So nori, that would wrap like sushi, that's actually an edible microorganism. And then as far as protozoans, hemolyphs, we'll be talking about those related to foodborne illnesses. And then fungi, just like algae, they're edible fungi like mushrooms. And so we'll talk about those both as contaminations but also as edible microorganisms. When we look at microbiology, we can break them into the different disciplines as far as application, again, like idea of food microbiology versus medical microbiology, but also the area of study of what type of microorganism you're focusing on. So each one of these, like virology for viruses, bacteriology for bacteria, have their own ology, okay? So for food microbiology, again, this kind of encompasses all these areas of study. So for food microbiology, we want to keep in mind that for most food, there's going to be one or more different types of microorganisms on it. And so it's really important that we keep that in mind because there's very few sterile foods that we ingest. And so the microbes that are present can play a desirable role in the food. So this is where we would use it for fermentation, but it can also have undesirable roles such as food spoilage and foodborne diseases. And so in order to study the role of microorganisms, sometimes when food microbiology is offered, there's a lab. So for our course and also due to COVID, we don't have a lab component to it, but we will definitely be learning about some of the different lab techniques. So there are ways in order to isolate the organisms that are found in food, again, either from a beneficial aspect or a contaminant aspect. And then we can study their morphology, so how they look underneath a microscope, physiology, biochemistry, so we could grow them on different types of plates and media and look to see what enzymes they have present, and also genetic characteristics, so we could actually do, look at their genomes and what genes they have present, okay? So there's many aspects with the lab component, and again, we'll look at some of that through literature, also some of the activities we do, okay? So some of the simplest techniques used in a microbiology lab, also in food microbiology, were developed, over 300 years ago, and so some of these basic techniques are, again, still used today, and some of them that you probably did in intro bio class. So if you look at the figure, you can see, you probably recognize a standard plate count, where you do the serial dilution, and then you put a set volume, 100 microliters, onto the plate, and you spread that out. And then after incubation, you can count the number of colonies that are formed. So for food microbiology, this would tell you whether you've met the threshold. So, if you have too many colonies, or certain colonies show up that indicate that you have a contamination, this is where you would then have to take steps to recall your food and to sanitize and clean the facilities to make sure that any of that contamination is removed. On the panel, or the figure that's on the right, has the multiple panels, you can see how there's cultural-based methods, and again, those are the ones you'd be most familiar with through the intro bio course. But we can also use techniques like molecular approaches, where we could do PCR and look at the genetic information, right? We can also do mass spec. So with mass spec, we can look to see what components are present, different chemical factors. And then we can also use electrochemical sensing methods. And so again, we'll be focusing on some of the more traditional techniques and some of the newer techniques that are utilized. So just looking at a kind of history of food microbiology, and looking at early history, we can't be sure, but there's probably a good chance that our early ancestors, the hunters and gatherers, were aware of food spoilage and foodborne diseases, right? So there's evidence that they used ice and fire to preserve or to cook food to make sure that it would be safe for them to eat. So there isn't evidence that they really were aware that there was causative agents that caused spoilage and foodborne disease. But again, they did take steps to make sure the food that was preserved and safe. Now, around 8000 BC, that's when we became a society that started doing agricultural, so we stayed in one location. We also had animals that we would raise. And so if we're having agricultural crops, we're going to have growing seasons. So there became a focus on being able to preserve the food when it was abundant, right? So if you've ever had a garden, you know there's that harvesting season, but there's a whole lot of time where you wouldn't have food. So you need to be able to preserve that so that you have a supply throughout the whole entire year. And so if we, again, look at historical sites, we can see that many different food preservation methods were being utilized. So things like drying like meats and fruits and vegetables out, cooking them, baking, smoking, salting, sugaring, using low temperature storage without air, so creating these pits that wouldn't have air to spoil the food, and then even fermentation, pickling, and spicing. So all of this was meant to reduce the spoilage, so again, that there would be food available throughout the year for people. So it might not have tasted the best, but again, it was better than nothing. And we'll talk more about later on about food preservation and how this actually controls for microbial growth. So this is one of the earlier artifacts that's been found that dates back to 5000 BC, and this is actually a jar that was used to store wine. So there was knowledge of how to transform something like fruit juice into wine. Again, not necessarily the idea of how it got there, how that transformation occurred. So in later periods, evidence through scripture, it's found that there was many regulations that were put on eating food. So the idea that you didn't want to eat meat that was from a diseased animal or one that was killed by a scavenger, so that was left behind where the carcass might have been rotting, or you didn't want to eat something that was handled by someone who was sick or unclean, right? And this was all, in a way, actually ends up safeguarding you from foodborne diseases, right? And so again, not necessarily this knowledge of what's causing the illness, but again, these steps that are being taken to make sure to preserve the food to be healthy and to make sure that people aren't becoming sick. Also, the process of fermentation. And so when we talk about fermented foods, we'll talk a lot about the cultural piece to it. So every culture has different fermented foods, and it was in part to preserve the food, but then there's also in that culture to make it desirable so it tasted better. And so if you've ever had one of those stinky cheeses, you may say, that's not very desirable, but for some cultures it is. And there's lots of different examples. There's like fermented fish, and where, in the American culture, it's not acceptable. It's kind of ugh. But in many other cultures, these fermented foods actually help to identify them, their culture. So the big turning point in the history of food microbiology, which really made it from just, preserving food, making it so it was edible, but actually understanding the science behind the transformation that happens with fermented foods, what was happening when we try to prevent food spoilage, is cell theory. So cell theory predates Hooke, but Hooke was kind of one of the first that's given credit to being part of cell theory. So Hooke was responsible for identifying cells underneath the microscope. So he built this kind of really rudimentary microscope and looked at cork under there, and he was able to see the cells there, and so he drew those. From there, Leeuwenhoek was able to observe single-celled organisms, so what we recognize as protozoans. So he took pond water, and again, he had a very basic microscope that he had built. And so he looked at those, and again, he did many drawings of different organisms that he saw, because there's so many variations with protozoans and what you could see in pond water. There were a number of other individuals between Leeuwenhoek and Pasteur. So Snow is important too. I usually talk about Snow in a general microbiology microbiology course when we're talking about epidemiology, because Snow and Florence Nightingale were really the beginning of epidemiology. They looked at the idea of transmission through a common source. So Snow looked at cholera and it being transmitted through the drinking water. So if you block a pump that's being used that's contaminated, you can actually stop the spread. Florence Nightingale looked at bandages. So they used to reuse bandages, and she found through her notes and record keeping that soldiers that were sharing bandages ended up having the same infection. So she said, hey, let's make sure they're sterile. Let's not share the bandage when we have a soldier that has a wound that has that's infected. Okay. And then we have Pasteur. So Pasteur did a lot of different things. He's known for pasteurization. And he developed that process because of contamination that was spoiling wine. So he, for food microbiology, he's a very critical person when we think about fermentation, production of alcoholic beverages. So you can thank him if you're having a glass of beer or wine. And then we also have Conch. So he looked at causative agents. So that's really important with the foodborne illnesses. Okay, so the ones that I've asterisked here are the ones that you should know about. Also, if you look at the PowerPoint down in the notes, there's a link to a microbiology textbook where this figure's from. So that has a whole chapter on cell theory and the different scientists. So you can find additional information about each one of these if you want. But again, Hook, Leeuwenhoek, Pasteur, and Conch are the ones that you should know when it comes to quizzes exams. So where are we currently in food microbiology? So when we look at current food microbiology, we can divide them into food fermentation with the idea of probiotics, food spoilage, foodborne diseases, and then this miscellaneous. And again, this is kind of how we're going to look at the semester throughout the course, is focusing on the current, what's going on in food microbiology, with giving you the background information also. So the next several slides just have a whole list of different topics that fall under these different current areas of food microbiology. So some of these we'll end up talking about during the semester, other ones we won't have enough time. So you can do a whole degree program in food microbiology. And so this course is really meant to be kind of an overview. What are the real key points, building upon what you learned in microbiology, okay? But with definitely with food fermentation, probiotics, the development of different strains, engineering bacteria that will give us these desirable metabolic activities, different products from that. Okay. With food spoilage, there's always identification of new spoilage bacteria. So as bacteria are changing and mutating, trying to identify them to prevent food spoilage from happening. So with that development of molecular methods to identify the metabolites of the spoilage bacteria, and ways to better predict potential shelf lives of foods, so that we can make sure that the food, when we're putting on a use by date, that is relevant. Also, this idea of quorum sensing comes in, because some of the food spoilage that happens really relates to microbial community. I mean, it's ecology and then quorum sensing. So probably at some point in the semester, we'll talk about this concept that you were probably introduced to, talking about biofilms in the medical microbiology aspect. And then with foodborne diseases, we need applications of molecular biology techniques so that we can have rapid detection of pathogenic bacteria in food and also in the environment. And then effective ways to detect and control for these pathogenic viruses. And within this, with foodborne diseases, there's the Puron diseases, which are protein-based diseases, which are very interesting. And so I never usually get to talk about them in the intro, so I definitely do want to talk about Puron diseases. You probably have heard of mad cow disease, and so that falls into that. There's also an elk wasting disease that's kind of become more prominent because there are cases of that in the U.S. And so there's, if you have like an elk burger, there's testing that has to go through to make sure that it's free of Puron diseases because there is transmission, there's potential transmission with that. So we'll talk about some of the Puron diseases when we talk about foodborne illnesses. And then miscellaneous, there's some of the different regulations that happens, and so we'll talk about some of those later on the semester. So hopefully this is a helpful overview of kind of what we're going to talk about throughout the semester, and introduces you to food microbiology and makes you want to learn more. If you do have any questions, please do not hesitate to contact me to answer those questions. And if there's any topics that you specifically do want to make sure we talk about this semester, please do not hesitate to contact me and say, could you please make sure that you talk about this? Because there's lots of flexibility in what we cover. And so even though I have certain things planned, I'm more than happy to make adjustments so that you guys are learning about things you want to learn about within the scope of food microbiology. Okay, thank you very much.